All right. So, good afternoon and welcome to Live at Lunch. Live at Lunch is a free noon hour lecture series with guest speakers on the third Wednesday of every month. And often the speakers, but not always, the speakers are part of staff here at the Royal BC Museum. So, it's a way to amplify and really celebrate the incredible work that's happening within the walls of the museum. And often, as we'll find out today, is the connections also with communities all around the province as well. So it's both within the walls of the museum and beyond the walls of the museum. So my name is Chris O'Connor, and I'm a learning program developer here at the Royal BC Museum. I'm honored to work and learn here on the traditional and ancestral territory of the Lekwungen people, known today as the Songhees and Swepsim, formerly known as the Esquimalt First Nations. And I imagine, Ken, because you're talking about other parts of the province, that you'll speak to the different territories and areas that you've been to as well. So research is an important mission of the Royal BC Museum. Staff and associates engage in subject-specific projects that help to build and better understand our collections. Today we'll hear from Dr. Ken Marr about fieldwork to the northern Alpine that has helped us better understand our plant distribution and what plants tell us about the last ice age, and many other things too. So Ken has a PhD from the University of British Columbia and has taught taxonomy at the University of Wyoming and University of Montana before joining the Royal BC in 2001. Please join me in welcoming him.
So now I'm allowed to speak, I believe. Thank you. So I was saying that I like to use this slide to acknowledge my father, from whom I learned a tremendous amount, not just about botany, but about how to be kind to people. And also I think this kind of summarizes kind of the nature of alpine and Arctic environments. And so that's what I like to start with. And yes, and we will talk about climate change. No, that's all right. We're going to talk about that. So as Chris said, part of our mandate here is to undertake research on the fabulous biota that we have in the province. And I came across this statement from a book by Francis Collins. Anyone know that name? He's Anthony Fauci's boss, at least when the NIH existed. Maybe it still does. So this is a really neat statement, I think, that summarizes science and research in general. And he's talking about his first chemistry class where the students were given a box and they're asked to guess what's inside. And after that experience, then he went into biology. But anyway, discovering what's in the black boxes that nature has placed all around us, and then letting your imagination and curiosity be further ignited by the candle you find inside. There was a candle inside his box. This is what science is. Using hypothesis generation and experimentation, science investigates nature in all of its beauty and complexity and discerns things that were not previously known. It's like a detective story, and that's what I think of whenever I'm undertaking science. Your observations and experiments provide clues about what the answer might be, though sometimes the clues turn out to be misleading, and you have to recognize that and regroup. Down deep, your investigation is built on trust. Trust that despite all this complexity, nature is orderly and adheres to regular laws. And persistence, just like in a good detective story, there will be an answer at the end. And with persistence, I had a feeling I missed a verb there. So kind of keep those thoughts in mind. And I think there's two approaches to science. One is like testing hypotheses, and another approach is just go and learn and see what you can discover. And so there's some of each here. So we're talking about Alpine. And as Chris said, I have been all over the province. I could not name all the territories that I've been in. But when I'm on the landscape, I am aware that I'm not the first person there. I am probably perhaps the first botanist making museum collections, but I think about the people who've been living there for thousands of years, how they lived on that landscape, without metal, without plastic. And I just imagine what their lives were like. So how do we define Alpine? It's called The Land Above the Trees. There's a book by that title. And globally, wherever mean July temperatures are less than 10 degrees C, trees cannot become established. So in terms of global warming, tree line is established not by any other factor other than temperature. So as temperatures rise, we can expect tree line to go up as well. So these are the places I've been. And you know, those are big dots, and it looks like I've been everywhere, but I think we're really just scratching the surface in terms of what's out there. And there's some areas we would still like to reach, the Columbia Mountains in southeast BC and the outer coastal mountains as well. So we've been making these collections since 2002. And the reality is that those of you who are familiar with the North understand that there's very few roads. And that distance across there between the Alaska Highway and the Stewart-Cassiar Highway is 450 kilometers. We're incredibly fortunate to have this amount of wilderness remaining. There's a few logging roads and mining roads that penetrate that area, but it's largely roadless and it's largely wild, allowing all the beautiful biota that we share this province with to continue to live. So this is some of the topics I'll talk about, and sort of in that order, and hopefully somewhat coherent. And I do want to talk a little bit about global warming causing climate change, and it's important that we link those two phrases because sometimes we talk about climate change or sometimes we talk about global warming, but global warming as a result of the greenhouse effect is causing climate change and short-term weather effects, as we're seeing with the polar air now reaching the southern states. So I'll talk a little bit about that. I often don't do that until the end, or if at all, because often we're asked, are you seeing any evidence of global warming and causing climate change? And I don't want to sensationalize, I don't want to overstate it, but the evidence is really, really compelling, and so it absolutely needs to be mentioned. So Richard and I started this project in 2002, started doing fieldwork in 2002. Richard is a paleoecologist. There he is digging up some sediments, and most of his career has been spent looking at sediments from lake bottoms and reconstructing past climates based on the pollen that's present in those sediments. And even though we're doing alpine fieldwork and collecting samples, he just couldn't resist, you know, collecting some soil samples. Whether or not he'll ever analyze them, I don't know. But he's a fabulous human being, retired without my permission before I was able to do that simultaneously. The other really key person is Will McKenzie, and part of what I was thinking about in this presentation is just the serendipity of some of the relationships we've developed and some of the places we've gone, and totally unplanned. So our first summer, and we arrived in Smithers, and Richard said, I think there's a guy here, an ecologist who's been doing alpine work as well. And so we went to the Ministry of Forest office and we met Will. And Will, turns out, he is documenting he's doing vegetation mapping of alpine ecosystems. We already have that for forest. Now he's been working on the alpine. So we teamed up, and it was really just the three of us for about 10 or 12 years. And he's a fabulous mountaineer, all-around adventuresome person. We'll see how long he lives. And then the other person who's been really involved recent years is Heidi Guest, who's the collections manager. And as time went on, we kind of added more people who were interested in working with us, and we were interested in them in part because they bring money. And so Mike Ryan, who's an ecologist in Kamloops, is a specialist on mosses, and so probably since about 2017, we've been working with Mike, and he's finding all kinds of range extensions, new species to the province, et cetera. Largely, a lot of work still to do. Probably my two favorite people are Darren and Claudia Copley. I call them Team Copley. They are they are definitely among the best naturalists in the province, and it's really fun Darren in particular knows birds and mammals very well, and it's really fun being in the field with him. A bird will go flying by, and Claudia will glance at him, and Darren will name it, and it's like, I don't know. I mean, the problem with birds, they move as far as I'm concerned, but he can discern that from the way its wings move, or the appearance or something else. So we began working with them in 2014, even though we're on the same floor. I didn't know they were working the Alpine in the, anyway, I don't know how we didn't know, but it's been a really good team effort. Another person who's been part of this group since about 2016 is a research associate, Rob Bennett, and he's a specialist on spiders. And so spiders are very poorly documented in the province. Through Rob and Darren, they've probably doubled the number of known spiders in the province. So these aren't charismatic megafauna such as grizzly bears or caribou, but they're every bit as worthy of our wonder and respect, and we should learn everything we can about them. What I didn't show back here was the manner in which these guys collect spiders is called pooting, not poutine, pooting. And they are, in their mouths, you see some surgical tubing, and they're sucking up little spiders. There's a baffle of cotton in there, and they suck them up, spit them into ethanol. And these are really tiny, and they're living under the rocks. Very brave folks. And so Claudia and Darren have also been collecting insects, and these are the various manners in which they collect them. The lower left-hand corner are surprisingly, to an insect, looks like a flower. And so Claudia adds a little bit of water and some detergent, which breaks the surface tension, and then the insects land there and donate their lives to science. And then there's more traditional ways on the right, collecting insects with nets. And the upper left-hand corner, that's kind of a passive structure called something. Does anyone know what that's called? I forget. Berlazy, thank you, Esther. Berlazy trap, and this is you set up in strategic places, often along stream sides where the insects are just trying to drifting along, and they drift along, they come into that tent, and then they go up into that cylinder of poison and donate their lives to science. And then Claudia pinning insects, very, very tedious, very careful in the work she does. Insects favor warm weather. So 2019 was a really poor year for being in the field. I don't think I took my toque off for day or night for about 15 days. And so here they are taking down their Berlazy apparatus in a snowstorm. So just a little bit, just acknowledge the things that they've been learning about, new species to science, lots of publications. They've been sharing the tissue with an organization in Guelph called the Barcode of Life, and they are getting DNA sequences from these animals, which then provides a whole lot more information. Another person that we've been partnering with beginning around 2016 is Shannon Birch, a mycologist, someone who studies fungi, in particular mushrooms. And this was an idea Erica Wheeler had, who used to work here, and she said, we're seeing these mushrooms in the field, why don't we talk to Shannon? And I didn't know Shannon, I knew of Shannon. And so we began collecting samples for her, and one year she came in the field with us. So part of her work is to take little pieces of mushrooms and put it in little tubes of ethanol, and then gets DNA sequences from that. But she also needs an image of what the mushroom looked like in the wild. And so take a picture of the mushroom on the right, and then there's my notes so we know where it came from. So this is what Shannon's been learning, and she's now working on a publication with a colleague at Kew in Britain to flesh out some of these new discoveries that she's been making. And there probably would be a whole lot more discoveries made if I hadn't left an entire box of samples on the mountainside in 2019, together with my sleeping bag, my wallet, and my Gore-Tex jacket, and my rubber boots. So a little bit about landscape history, and just to understand the place where these plants, and I'll mostly be focusing on plants now, where these organisms live. So if we think about our province, the prevailing view is it was covered by ice multiple times during the Pleistocene, and this is what it might have looked like, a giant ice sheet and maybe some little mountains sticking up above the ice sheet. What we don't think about is what did it look like after the ice melted, and this is what it looked like. I wasn't there, but this is, there's no vegetation, there's no soil, no animals. It's just a totally, maybe almost totally sterile environment. And so we don't think about that when we walk around in the forests and all the other places we go. So if you go from west to east, west to east across the province, we still have lots of glaciers. The coast mountains, if you fly north, you will look out the window at the spectacular glaciers. And then we go in more into the center of the province, largely for the most part, there's no glaciers remaining, but you can see those little, where little glaciers have eaten out some of the ridge line there. So there's still evidence of glaciation. And then you get into the northern Rockies and you get into great big glaciers again. So the landscapes that are left by glaciers are, one is broad U-shaped valleys, river-shaped valleys, or V-shaped glacial valleys, or broad U-shaped valleys. And they leave other features on the landscape. So think of a glacier as a conveyor belt. And during full glacial times, the temperature is fluctuating quite a bit. So maybe nearly every day, even during these cold times, the temperatures get above freezing, water seeps into the cracks in the rock during night, freezes. What happens to water when it freezes? Expands. Okay, so it cracks. You get all these cracks, all these rocks falling on the ice, and then they're getting transported because it's moving by gravity. And then as those rocks are in the glacier, they're getting ground up, and finally when they melt, they have this glacial silt. But they also leave on the margins of the glacier what's called a lateral moraine, and that's what that feature is. And then on the left, that's an arête. That's a glacier just been eating back at the ridge line, leaving this really sharp feature. There's also such a thing as a rock glacier, and if you know what these look like, you can start to recognize them on the landscape, even on Google Earth, which is my absolute favorite. I don't know if it's an app or whatever it is, but it's one of my favorite things to understand the land better. So this is it's a core. It's got an ice core, and it's just flowing down the mountain, and you can see those kind of ripple marks there. So that's not just a talus slope. It's actually a stable talus slope. It's actually moving. Another feature you may or may not have noticed when you're in the Alpine is something called patterned ground. And there's this somewhat mysterious, maybe Noemi, and you can explain it better than I can, but there's this mysterious process whereby rocks, you start out with a homogeneous mix of various size rocks, gravel, fines, and the rocks get lifted up, freezing underneath, lifts them up, and then they form these kind of circles. And vegetation can't get it, and you get a sorting of fine materials in the middle and then these coarse materials on the outside. And there's almost a predictable diameter and almost a predictable size of the largest class as well. So vegetation can't get established as long as that freeze-thaw process is happening. But after a while, something changes in the soil moisture or the temperature regime or something, that those central portions become stable, and so you do get vegetation. And even on a really small scale, you can see this process at work. This is the bottom of a pond. Oops. One other feature I've only seen once is these unusual mounds that have an ice core to them. So somehow the groundwater is moving along, and in various places it just comes up. And all the way down that valley, you can see those little mounds. And they're called palsas, and in the Arctic they're much bigger, called pingos. But I've been told this is a palsa. Global warming causing climate change. What do you see in that meadow? What do you not see in that meadow? Don't see tall trees. What else do you not see in that meadow? Dead trees. So that's a forest that has recently arrived. And you project 40, 50 years from now, each one of those trees is growing that much laterally, maybe that much vertically. What's going to happen to that meadow? If the process continues, it'll no longer be a meadow. It'll become a forest. And here's where, this was really astonishing. So Darren and Claudia and I got dropped off on this ridge top, and there's these trees at 2,000 meters, but the actual tree line is down at 1780 meters, as that shows. And they're growing along pretty well, actually. Yeah, the vegetation is changing as a consequence of global warming causing climate change. So here's a really maybe beautiful view, kind of idyllic. Oh, isn't that a nice place? Look at that. And, there's their tents at the bottom of the rainbow. But it's actually quite a bit more serious than that. And you see the vegetation there in the front. Those are clumps of vegetation that are just falling off the ridge, off that slope, as the permafrost melts. So here you have maybe a really old moraine, some kind of deposition, and it's just melting. And these chunks of vegetation are just sliding off. And based on the fact that that's a north-facing cirque, so the growing season's very slow, not a lot of growth each year. It takes a long time for soil to develop. And so those chunks of turf are probably thousands of years old, and they're just falling off. And I didn't really realize it. Initially, I was walking along these big cracks in the turf, and I was puzzled by that, and then I saw the rest of the story. Oops, that one's a little off. Yeah, so glaciers are melting, and this is a photo I took in 2016. So what you see there, this is August 2016, a lot more warm days coming up, and very little of that glacier is protected by snow. So in more stable climates, every year ice accumulates thanks to the deposition of snow during the winter, and then much of the glacier is protected from the sun during the summer months. But this glacier, I can't imagine it's still there, and it was already just a remnant. This is looking down the ridge from that right beside the top of that glacier. And so imagine what are going to be the consequences for that watershed when that glacier, which is essentially a water reservoir, when that melts and is no longer releasing water into the system. You can imagine what the consequences will be. So I was in Dushanbe, Tashkent, Tajikistan in October. And in the National Museum there, they were proclaiming the International Year of the Glacier, which is this year now. And in Central Asia, they are very, very aware of the seriousness of loss of glaciers because, as I said, it's a water reservoir. And agriculture and domestic use, and to say nothing about all the other biota, depend on that water being released. So this year is International Year of the Glacier. A bit of a personal story. So this is a bit of a, well, it's a serious tragedy in that this Swiss glacial scientist he had spent 30 years on a research station on the Greenland ice cap, and he fell into a crevasse. And part of the tragedy is that his son was there. Another part of the tragedy is that he had just said, he was in their group facility of some kind. He said, I'm going to my tent. And so everyone thought he'd gone to his tent. And then hours later, they discovered he wasn't there. And so he must have wandered off, did something different. The personal aspect to this is my mother's neighbor in Boulder, Colorado for 20 years. He was a professor there. He was also the Swiss consulate. And then he was enticed back to Switzerland to manage a research institute of 600 people. And the government wanted him so badly that they decided to allow him to exceed the mandatory retirement age. So a really fabulous guy. Had a number of meals with him when I visited my mom, and he used to come and shovel her snow and clean out her gutters. So very sad, but I do want to honor his memory by mentioning that. So field work. So I think I mentioned earlier, access is really difficult because these mountains are so remote and there's very few roads. We've hit a few summits on roads, but we want to get to places where no collections have been made in the past. And so one way to do that is a float plane. Float planes are about half the cost of a helicopter, but a float plane needs a kilometer length of water to take off 3 sort of heavy guys. So they won't land you somewhere where they can't come back and pick you up, which is good. It turns out, each year when we're planning field work, I try to find lakes that would be the right size, and there just aren't very many of them. But this year we were able to use a lake. And, if by chance a boat falls out of the sky, then we use the boat as well, especially if it has fuel attached to it. But most of our access is by helicopter, which is pricey for sure. And we've been fortunate to have funding from various sources, not least is the Royal BC Museum. So a helicopter can get you places that you're never going to get to otherwise. Days on horseback, if then, certainly a long, long way to walk. Fabulous machines. The best way to use a helicopter is to, once we establish where we want to camp, we throw out our gear and maybe Richard and some other people, and then hop back in the chopper and get flown to the nearest high point. And by doing this, we're able to maximize the helicopter. And in a very short time, it can fly you quite a ways away. So this is the start of a field day. Reasonably good weather in Atlin Provincial Park in the northwest corner of the province. And then that's where our camp is, and Richard and Rob Bennett, and maybe another person or two. So we spend the first day collecting all the way back to the camp, and then the next 2 days collecting in the area around camp. And our goal is to collect specimens of every single species we encounter on the landscape in 2 or 3 days. So this is a kind of a somewhat typical scene. Everyone with their own tent. It's just better in bad weather especially. But in early years, we did double up some because space was restricted in the helicopter, especially the smaller choppers we were using. We do get some nicer campsites as well, nicer weather. This was last summer. And you can see actually there's a lot of bedrock, and so it wasn't easy to find a good place to set up a tent. But that's why we're all spread out there. And we'll introduce these shelters to us, just fabulous places to shelter. It doesn't have a floor, but we set up one for cooking and to keep the food away from our tents because we are always in grizzly territory, and another shelter for processing specimens. So we're visiting every habitat on the landscape as we understand it, and we're really pretty intentional. There's large areas that are pretty homogeneous, and then there's areas, moist areas, ridge tops that have a little more specialized vegetation. And so as you can see here, just looking at the different colors where those arrows are gives you a sense of how diverse the landscape is, going back to the first slide where my dad talks about in relatively short distances, you get some very different environments. A lot of this has to do with snow deposition, or I should say redistribution by wind. So the snow falls evenly everywhere, and then the wind redistributes it. And when that melts out in the summer, then you get these different patches of areas of high moisture and low moisture. So a typical day for me, wandering all over the landscape, trying to hit as many different habitats as possible. And it's a good workout. It's exhilarating because we're in places no one has made collections before. And I'm really careful to say no one's been before, because we're certain indigenous people have been on this landscape for thousands of years. And some of the plants, you'll only see them once. They're only in one place. And so you just have to cover the territory. And obviously with Heidi and sometimes other botanists, we can cover the ground a little better. Just what it looks like on the ground. This is starting off the day working with Will, and he's doing ecological plots. And so his goal is to spend a lot of time in one place. And so we'll work together documenting all the plants there. And then I want to move on. Once I've spent 5 or 10 minutes somewhere and I think I've collected everything, I want to go somewhere new. So I ditch him, but we stay in touch. And later on, rejoin him. And so here we've gone from a really a windswept, dry meadow, and then down into a really wet meadow. And every one of these places have totally different suite of species. And then there's Mike taking photos for iNaturalist. So we collect our specimens. We used to just put them in bags and drag the bag around during the day, which was really not great on specimens. And then Erica Wheeler said, you know, there's something called a field press. I said, oh, and actually my mother had made one for my dad, but I'd forgotten that. So the field press is really good. So we carry this, it's kind of like a satchel. You can spread out the specimens on the newsprint, and then it folds it up and you tighten it up so that they start to begin to press, and it stabilizes them as well. And then this is what it looks like all bundled up. The screwdriver, I've nicknamed it a plant extractor, but it is a screwdriver. And they're a lot cheaper than Leatherman tools. I think my first year I lost 3 $50 Leatherman tools digging up specimens. So, you know, $8 or $9 screwdriver. I've left a few of those on the landscape as well for future archaeologists that will really be puzzled. So the standard thing for anybody doing field biology is to have a notebook, writing down every stop. You record the latitude, longitude, elevation, related vegetation, soils, wood that I knew more about soils. And so anything that someone 50 years, 100 years from now will want to know about these specimens, because we're collecting for the present as well as for the future, as Chris mentioned. We're all carrying radios. We check in every 2 hours. Everyone's, you know, where are you? What's your plan? Sometimes we're together, sometimes apart, but it's just a safety feature. And then GPS for obvious reasons. So at the end of the day, then transferring the specimens from the field press into the more proper is the wrong word, firmer plant press with wooden frames and strap. And on occasion, on a family holiday, I've taken advantage of the opportunity to make collections as well. And here's my dear daughter, who is now 26 and fell very far from the tree. But she loves to be outside, and she's doing a PhD in particle physics at SFU. And where that goes, I don't know, but she loves it, and she loves to be outside. She has a wonderful collection of houseplants. She could care less about their names, evolutionary relationships, but that's all good as long as she wants to be outside. And yeah, I worry about her actually sometimes. Ooh, anyway, I won't go into that. So then getting our specimens dry is a big challenge because here at the museum, if we make a collection nearby, we have a cabinet in the basement that has expanded sheet metal and so a fan in the bottom and a heater. We can blow warm air up through the cabinet and in 2 or 3 days get the specimens dry. These techniques here will not get specimens dry in probably 2 or 3 weeks, but we're doing the best we can. So the consequence of that is we needed to be taking a pretty significant bulk of pressing supplies with us in the field, and sometimes we're out 5 or 6 days at a time, filling about that much volume every day. And so getting the specimens dry is an issue, and space is limiting in the chopper. So Colin Longpre and I, several years ago, I kind of said to him, can we build something that we could take with us in the field? And Colin did. And so it's a box that collapses, and the lower right-hand corner is the original prototype with 4 sides. Coleman makes a flameless heater, and so we figured out the optimal height, obviously, because that's all paper above the heater. And they stopped making them, I think, maybe too many accidents with them falling over in tents or something. So when we realized that, we went on Use Victoria, and we bought 2, so we have 3 now. So very grateful to Colin because he saves us a tremendous amount of bulk, and we get our specimens dry in 18 to 24 hours. We can take them out of the press, bundle them up, and do the next day's specimens. And every helicopter pilot wants to know, what is that thing? And Darren and Claudia are tired of me explaining it, but you haven't heard this before. So, and it's just wonderful. I've set it up in motel basements, out in the field here, and there's Erica Wheeler, who used to be a collection manager here. So the second one that Colin made was has piano hinges, so it just folds up. Before, like the 4-sided one, trying to stabilize it on a uneven ground in the wind was not optimal, and the next generation is much lighter. So what happens to all these collections we're making? They get processed, and that involves, we have some wonderful volunteers, some of them are here. who mount the specimens. That involves gluing or taping them to acid-free paper. And then they get databased by me, by volunteer, and then they get filed in these cabinets. And this is an old picture of John Pendermoss, who was the first collection manager. Did anyone know John? You guys did. Did you know John? Yeah. Yeah. So, yeah, that's what happens to them. So how are they used? And actually, it's really important to understand that all of our collections, any collections here, not just plants, they're constantly the information associated with them is constantly being accessed. And due to all the online access people have, we don't even know anymore. It used to be before there was online access, if someone requested a loan, you knew so-and-so was working on the specimens. We can't even know that right now. But specimens are used to document biodiversity, and a lot of the species descriptions require an actual specimen, and then they want to know, okay, so you've described this new entity, what collections did you use to base that on, so that in the peer review process that can be evaluated. So here's a new species of potentilla, and there's highlighted in yellow collection we made. Here's a really cool study. I'm part of a Western North America Alpine botany nerd group. And so every month we talk about a paper, and this is one we talked about a year ago, and a group, including one of the nerds, looked at species diversity. They actually looked at phylogenetic diversity, but let's just call it species diversity, from the southern tip of South America all the way into the Arctic. And they accessed their data off of this facility called GBIF. And since we share our data with GBIF, at least some of it, probably in this analysis, our specimens here have been included in that analysis. Especially during the 2000s and even more recently, there's a group of northern botanists really interested in tracing the migration of Arctic and alpine plants. And there's a group in Oslo, and then there's Hajime Ikeda in Japan. And so he's been looking at this particular plant. It's a type of harebell. And we had taken it as far as we could with our analysis, and actually we didn't find anything. But he had better tools of DNA sequencing. And so because so many of Arctic and alpine plants extend all the way around the northern hemisphere, and so he wanted tissue samples, which we had, and so we were very glad to help him out with his analysis. So those different colors of dots are different genetic types. And he did come with us in the field one time, and he spent, I don't know what happened. The second day he spent the entire day in his tent. We didn't know if he was alive or unwell or what, jet lag, just came from Japan. I think partly he was just overwhelmed. Like, we get dropped off and we're just walking across the tundra. And we did see wolves that day and a caribou. Anyway, but he's been doing really good work. We do share a report with First Nations, whoever's territory we're in. Kudos to Claudia, who does a lot of the formatting. So this is from Mount Robson Provincial Park. It's hazy in the background from forest fires. And so we give them a pretty thorough species list, and then we try to highlight the various things that we think would be a little more interest than just this is all the plants we saw, but what are some of the interesting stories associated with those plants? So around 2016, we were stuck in McBride. We couldn't fly because the weather was poor. And so Erica and Will and Erica mostly said, you guys have to do something more with this data. And I was kind of resistant. I don't know why I was resistant. But anyway, I was resistant. And she said, no, you've got all this data. You need to do something more with it. And so Will is very skillful with data analysis. And so what we've done is a geospatial analysis, which involves comparing the list of species from every mountain with every other mountain. And as you would expect, the mountains that are closest to each other have similar flora. But what this does tell you is where are the boundaries between those clusters with similar flora. So the next question is, what is the reason for that? So that's ongoing work. Some of the adaptations of alpine plants, and maybe this is already familiar with you, but just keep in mind the extreme temperature changes. So they're living in a different world than here at sea level. And there's a very short growing season, sometimes as short as 6 weeks. So cushion growth form is really adaptive, and many different lineages of plants have found that as if they're thinking, okay, they're thinking. It's better to be small and stay close to the ground and contain the tissue within a cushion. And so it retains moisture and it reduces the damage from wind. And in a short growing season, there's really no advantage, at least in those windswept sites, to getting tall. So they're able to produce flowers very quickly. Moss campion, which, just to be clear, is not a moss, although it may look like it. And you can see the cushion form there. I did sacrifice that plant for this photo. And this is one the mounters will tell you is their least favorite to mount, probably because they're so thick. So I've considered maybe this is British Columbia's most important plant, maybe all of Western North America. And why is that? You can see here a big cushion about this big, but that cushion was established on all the plants growing on it. There's probably 8 or 10, maybe 12 different species of plants and lichens and mosses, but they all got established because that initial cushion was there. Absent that cushion, you can see around it, and in this image, you can see there's almost nothing growing. So, and sometimes I find that species in lush meadows, and so I try to imagine, okay, I don't think that species was able to colonize that lush meadow. I think it's a remnant from the initial colonization event. Short stature, this is probably the second smallest alpine plant, easily overlooked. So most plants are perennials, due again to the short growing season. There's no guarantee, for annual plants, there's no guarantee that they're going to be able to complete their life cycle in the short growing season. So most plants are perennials. And here's a slight detour, use of our specimens. So there was an honor student, Vic, who he did detailed measurements of the leaves from our collections. And what he discovered was that there's this taxonomic complexity. What he discovered is you have two species and then you have hybrids. And the hybrids actually have a range that's quite a lot greater than one of the parents. And there's a couple of reasons for that. But this is kind of unusual. It's not at all unusual to find hybrids in plants. Lots of phytopanchy going on out there, especially if the chromosome numbers are the same. But to find the hybrid having a different distribution from one of its parents is unusual. So just a few highlights. So even talus and fell fields have vegetation. And from a distance, you look out there and you think, there's nothing there, no point in spending time there. Wrong. There's lots of plants. And this is one of my favorite. It's really early to bloom. And so we rarely catch it in flower. I don't know if it's in cultivation or not, but it should be. It's spectacular. And these, maybe some of you grow monkshood in your gardens. This is a tiny little version of that, about this tall. And as you get closer to tree line, the plants get taller and they have more flowers. And again, notice that big hummock there, that big cluster of vegetation. Soil's been trapped by the moss campion. Dreadful drabba. So this may take us over time a little bit, but I have to read this to you. So this is the best book to learn about alpine flora by McKinnon and Pojar. Andy McKinnon has talked to us about fungi before, and it was the Green Party candidate. So the thing that makes drabba dreadful is their classification is based on these microscopic hairs. And its classification has to be based on genetic features, and that's a very good genetic feature. It's just hard to see. This is what Andy and/or Jim wrote. I suspect it's Jim. Drabba is indeed a difficult genus to deal with. The radical honesty self-improvement movement, spearheaded by psychotherapist Dr. Brad Blanton, advocates telling the truth, always. Even if you're asked by someone for whom you care deeply, does this kilt make me look fat? Such frankness can be disarming, charming, or alarming, but is said by proponents and converts to result in better communication, improved work environments, and stronger, more loving relationships. In the case of drabba and your whispered question, does it really matter what species of drabba this is? No, it doesn't. Not to the plant and not to you. Unless the species is rare and endangered, you are trying to save it, and correct identification is crucial. Now, of course, it can be fun and gratifying to work your way through the maze, but sometimes it is more satisfying merely to acknowledge the beauty and complexity of these alpine gems. So you gotta love these guys, right? They would put that in this book. So some of the things we have found, fortunately, we have found some species that were thought to be rare, and because not a lot of exploration had happened, we're able to say, no, not so rare. So this is a beautiful pale poppy. We found several new populations, and so it's been downlisted from the rare list to the less rare list. Really common habitat, especially in the south, is these dry meadows and heath. And here's a really puzzling thing that stumped me for quite a while. And to make good collections, you should collect flowers. And so I was often encountering this plant and it didn't have flowers. And this is annoying. So I'm crawling around, walking around looking for specimens with flowers. And then finally, I started realizing what I'm seeing here is a fake flower. And I would not have known that, except that my dear mother sent me a similar situation where this plant here that has those fake yellow flowers, it should have a tall stalk with white flowers. And so that is exactly what this is. It's a fake flower, and it's capable of producing nectar, and presumably attracts insects, which then disperse its spores. But nobody has actually investigated this yet, as far as I know, and I've searched the literature. So there's a name for the fungus that causes that infection, that causes the leaves to have that unusual growth form. And yeah, lots of images, but nobody has actually sat down in a meadow and spent some time watching, are there any insects that come there? And so using our collection, actually, I was able to, you can detect this on herbarium specimens, so I was able actually to document the distribution of these false flowers, and they occur on 2 different species, closely related, all over the province. So here's another situation. The flower on the left is female, the one on the right is hermaphrodite. And plants, because they can't move and choose their mates, in the course of evolution, there's been many different mechanisms by which plants can prevent self-pollination, self-fertilization. So this is one of them. The flower on the left, the female has to get pollen from a different individual. So all populations, in order to persist, must have genetic diversity, maintain genetic diversity. Moist meadows, so a bear in a very short amount of time turned over the turf there, looking for roots or perhaps small animals. We do occasionally see places where bears have tried to dig out a marmot, and it's a fearful thing to see because they can move big boulders and create great craters, and you don't want to be around when they're doing that. There are some edible plants in the Alpine. This is one of them. Has a kind of a crunchy, nutty flavor. Some plants flower, but the flowers are sterile, and so this is an example. There's limited genetic diversity in this species, so sometimes it must be crossing, but mostly it reproduces by little bulbils. So the stalk on the left, those are tiny little plants that just sprouted right on the plant from these vegetative buds. There's no fertilization, pollination, fertilization, or seed formation in this. It's just, it's kind of like the same thing as a strawberry runner, just reproduces vegetatively, which is a big advantage in an environment where pollination is not certain. However, the offspring are genetically identical or nearly so. In the back of this flower, you see this bee, and that bee, that's a bumblebee. It is not going to get into that flower. So what's it doing? And is actually it's robbing the nectar of that flower, and it's chewed a hole in it, and it's getting the nectar out of it. So one of my favorite groups are the louseworts. Some of them are buzz pollinated. The insects just hover and they vibrate their wings, and the pollen falls out onto their bodies. They don't actually come in contact with it. And then there's others which insects do crawl into. I'm going to skip that one. Runnels and wet meadows, lots of lush habitats with melting snow. Bog orchids. If you encounter this one, you may smell it before you see it, and it grows in the ditches around on the peninsula as well. It has a wonderful fragrance of cloves. So if you're smelling cloves, this guy is in the neighborhood unless you're in your kitchen. Here's a real puzzle in terms of its name. A Schultz's Little Nightmare. I don't know how it got that common name. It puzzled me for a while as well, but I didn't experience a nightmare. And so we do report new finds to the Conservation Data Center, which is the CDC. We are their repository for collections. Yeah, and so we found thousands of plants, significant range extension. This is probably not because of the plants are expanding the range, it's probably because nobody's had the opportunity to go look. Distributions. I'm gonna skip a little bit there, although I would love to tell the story associated with those last two slides, but we don't have time. So ice grass occurs in the northern part of the province all the way to the Arctic, and then occurs in the Beartooth Plateau, and then again the mountains of Colorado. And so what's that distribution about? And it turns out there's about 17 species that have a similar pattern of distribution. You can probably eliminate the constipated bird hypothesis. And so there's a real puzzle, and this is kind of the kind of information that you can get from distributions, not only from our collections, but from collections from other institutions. So there is a view that all of the province was covered by ice during the last ice age, multiple times, as I said. And in 2001, Richard, I was in his office, and he said, I don't believe the province was covered by ice in the last ice age. And I said, oh, I thought that was pretty well established. Nope, I don't believe it. Richard doesn't mind being outspoken. So the theory then would be that after the ice melted, BC was colonized from the south and from the north by plants and animals coming into the province from areas that had escaped the ice age. So now we've been using DNA markers to trace the migration of Arctic and alpine plants, just as this has been used for our own species. And this is the first plant we studied. And it's probably the most common Arctic and alpine plant. It's in the Himalayas, it's in the Pyrenees, it's all over North America. Wherever there is alpine habitat, or has been, and not too long ago it occurs. We did get funding to go to southern Siberia and to far eastern Russia to collect tissue. To make this study kind of valid, you need samples from outside the area as well as within, so as big a geographic region as you can get. Here's a wonderful woman, Marina Olenova, a botanist at Tomsk State University in southern Siberia. Didn't hear from her this year for reasons I guess we can imagine. But every year for years, she would send us these lovely Christmas cards of winter scenes in Tomsk. So the way this work is done is grind up the tissue, extract it, put it in this box, this machine here, we call it a PCR machine. This is the polymerase chain reaction where that takes place. Everyone learned about that during COVID, right? The PCR test. Well, this is the equipment that's used, been in use since the early 90s. We send the tissue to a lab in Korea after using an enzyme that comes from hot springs. So all the molecular biology depends on an enzyme from hot springs and or deep sea vents. And then you get a printout online, the peaks, and then you get the DNA sequence. Then you can compare DNA sequences to each other. These are chloroplast regions, and these are non-coding regions of DNA. So a change in a non-coding, they used to be called junk DNA, doesn't really affect the survival of the organism. And so you can trace relationships. So you take a whole lot of DNA data, and you assemble it together, and you get what Richard calls a spider diagram, which is profoundly unscientific. This is a haplotype network, is what it is. And so this gives you a sense of the relationships among all those different samples. So every gap is a single genetic difference, a single DNA difference. So that has some meaning to it, but then you need to plot where do those occur on the landscape. And so here, as mentioned earlier, there's a group, forget the colors and the shapes, they're all one group, that came from the north, and we did get samples from those green dots or yellow, whatever they are, in the Arctic. We did get samples from other people. So that lineage came from the north, as predicted. Here's a lineage that came in from the south. as predicted. But then there's a whole lineage that only occurs within the borders of British Columbia and south, and there's a big gap there. And this is the area that was presumably covered by ice. So we think this is somewhat evidence for those areas, perhaps somewhere in that area, escaping the last ice age. And then, as it turns out, there was a group in Scotland and a group in China studying the same species, so we sent them our samples. They did some really complex analysis, and I got my name on the paper. I don't really deserve it, other than the fact that I thought of analyzing the same plant, and so I made collections, but their analysis is really complex. So what they basically showed is this species originated in the Himalayas. It went into the Arctic, it came around, and they joined up in Greenland. All of that information would be totally unknowable if not for DNA. So just to wrap up real quickly, so our idea that these areas were not covered by ice is getting more and more support. Here is an area in the northern Rockies where thin-horned sheep molecular data also supports an ice-free area. And sorry to Heidi, I'm going to have to skip that. And I'm going to skip these two. So what have we learned? We've learned a whole lot that we wouldn't have known otherwise. It's been a really rewarding project. I'm really proud of what we've accomplished safely for 22 years in the field. My two favorite quotes, these are from my wonderful folks in the back. If you don't go, you don't know. I mean, that's just, we have learned so much. And if we hadn't gone, we wouldn't have learned it. And then I love Brittany's response to the question. And people ask, what's your favorite plant? Well, I like Brittany's response, whichever one is blowing my mind. Lots of people to thank. Laboratory assistants were fantastic. Got funding from a bunch of places. Tech Industries allowed us to use one of their camps. Best food ever. Laundry, somewhere to sleep. So now I got like 3 minutes of just, when you have more pictures than you have time, it'll be timed slides. Sorry for going over. Get up and leave if you'd like. But this is the statement that was at the top of the escalator in the museum when I started here. And I think whatever terms you want to use, vision, mission, whatever, management speak, I think this is one of the reasons why we exist, and it's important to keep that in mind. Chris, for going over. I knew I would too. Oh, well. Thanks for coming. As public servants, I think it's really important that we tell you what we're doing. You may or may not think it's important what we're doing, but we're working really hard at it. So thank you very much for coming.